Life, in all its magnificent diversity, is united by a single, elegant molecule DNA. This double helix holds the instructions for building and maintaining every living organism on Earth. From the smallest bacterium to the largest whale, DNA serves as the blueprint of life. Each rung of the DNA ladder is composed of two chemical bases, paired together like puzzle pieces. These bases, adenine, A, thymine, T, guanine, G, and cytosine, C, form the letters of the genetic alphabet. The sequence of these letters, known as the genetic code, determines the specific instructions encoded within DNA. Our understanding of DNA has revolutionized the way we view the natural world, offering new tools for diagnosing and treating diseases. The Earth is teeming with an astounding array of life forms, from microscopic bacteria to towering trees. This diversity is a testament to the power of evolution, driven by natural selection. Charles Darwin's concept of natural selection describes how organisms with advantageous traits are more likely to survive and reproduce. Over generations, these small changes accumulate, leading to the evolution of new species. DNA, the molecule of heredity, plays a central role in this process. Occasionally, errors occur during DNA replication, leading to changes in the genetic code. These changes, known as mutations, are the raw material of evolution. Some mutations can confer a selective advantage, making the organism better adapted to its environment. One of the most fascinating aspects of DNA is its ability to replicate itself. This process, known as DNA replication, ensures genetic information is passed on from one generation to the next. Occasionally, errors during replication lead to the duplication of entire genes or larger segments of DNA. These duplications can have profound consequences for evolution. Duplicated genes can accumulate mutations without immediate consequences, exploring new evolutionary possibilities. Over time, these duplicated genes can acquire new functions, leading to novel traits and potentially new species. Mutations are the driving force of evolution, providing the raw material for natural selection. These changes can be as small as a single letter in the genetic code or as large as the duplication of an entire chromosome. Beneficial mutations are more likely to be passed on to future generations, increasing in frequency within the population. Mutations can arise from errors during DNA replication, exposure to radiation, or certain chemicals. Some mutations are spontaneous, occurring randomly without external influence. The rate of mutations can vary depending on the organism's environment and its DNA repair mechanisms. While essential for evolution, mutations are a double-edged sword, balancing between too few and too many. The diversity of life on Earth showcases evolution's creativity. From butterfly patterns to bird songs, evolution has produced astounding adaptations. Consider the evolution of snake venom, a potent cocktail of toxins evolved from harmless saliva. Mutations in genes coding for salivary proteins gradually transformed them into deadly toxins. Over millions of years, these toxins evolved to target specific physiological systems in prey. The evolution of snake venom exemplifies how mutations can lead to novel traits. This process has driven countless other adaptations, from chameleon camouflage to deep-sea bioluminescence. For decades, scientists have puzzled over vast stretches of DNA with no apparent function, often called junk DNA. These regions make up a significant portion of the genome, far exceeding the DNA that codes for proteins. Recently, researchers have uncovered surprising functions for these sequences, suggesting they play important roles. One possibility is that junk DNA regulates gene expression, acting as switches to turn genes on or off. Another possibility is that junk DNA helps organize the DNA molecule within the cell nucleus. These sequences could act as spacers or help fold the DNA into complex structures. The origin of new genes has long been a central question in evolutionary biology. New genes can emerge from non-coding DNA, once dismissed as junk DNA. Random mutations can create new start codons within non-coding DNA, potentially transforming meaningless sequences into functional genes. Most such mutations are neutral or harmful, but a small fraction can lead to beneficial new genes. These new genes, initially crude, can be honed by natural selection over time. This process gradually increases their complexity and sophistication. Evolution is an ongoing saga, shaping the destiny of life on Earth. Every organism alive today is the product of billions of years of evolution. As environments change, new selective pressures are placed on organisms. Those with advantageous traits are more likely to survive and reproduce. This process of adaptation and change is happening all around us. Bacteria are evolving resistance to antibiotics, insects to pesticides, and weeds to herbicides. The evolution of antibiotic resistance is a particularly alarming example, creating a growing crisis with potentially devastating consequences. As we delve deeper into the genome, we gain insights into our evolutionary history and the forces shaping our species. Powerful technologies like CRISPR-Cas9 allow us to edit the genome with unprecedented precision. This technology has the potential to cure genetic diseases and enhance desirable traits. However, these advances raise ethical concerns, such as editing the human germline and the potential risks and benefits. We must proceed with caution and engage in thoughtful public dialogue about the ethical implications of genetic engineering. The ability to edit the human genome poses significant ethical challenges. One concern is the potential for genetic engineering to exacerbate social inequalities. Limited access to these technologies could create a genetic underclass. Another concern is the potential for unintended consequences, as our understanding of the genome is still incomplete. The use of genetic engineering for enhancement purposes raises questions about social justice and fairness. We must navigate this uncharted territory with caution, guided by ethical principles and a recognition of the potential risks and benefits. DNA, the molecule of life, is a testament to the elegance and complexity of the natural world. This double helix holds the instructions for building and maintaining every living organism on Earth. Our journey into the world of DNA has revealed a universe within, a realm of astonishing complexity and beauty. We have witnessed the power of evolution to shape life over billions of years. We have marveled at DNA's ability to replicate, mutate, and evolve, giving rise to life's diversity. As we explore DNA's mysteries, it will continue to surprise and amaze us, revealing new wonders. The journey of DNA is a story still being written, a testament to life's ongoing creativity and resilience.